this video discuss about biocontrol agent of Bacillus subtilis. General characteristic of Bacillus subtilis: gram-positive bacteria, rod-shaped, arrangement single, pair or chain, size 4 to 10 micrometer in long and 0.25 to 1 micrometer in diameter. Family Bacillaceae. Motility motile, swarming motility. Capsule absent, endospore present. Respiration facultative anaerobic. Optimum temperature 25 degrees Celsius to 35 degrees Celsius. Optimum pH 8. Bacillus subtilis also called the A bacillus or grass bacillus. The bacillus refer to shape of the bacteria like rod shaped. Subtilis means slim or slender. Okay. Bacillus subtilis bacteria secrete enzymes such as the amylase, protease, pulinase, citinase, silanase and lipase. This enzyme produced 60% of industrial enzymes. Next, in the history of the Bacillus subtilis, previously the Bacillus subtilis was known as Vibrio subtilis. The bacteria discovered by the Christine Gottfried Enberg in 1835. In later renamed the Bacillus subtilis 1872 by Ferdinand Kohn. The main habitat of endospore forming the Bacillus organism in the soil. The most Bacillus subtilis in soil environment and plant growth. Bacillus subtilis as a plant disease control agent. The numerous species of the Bacillus identified in a PGPB, plant growth promoting bacteria, and biocontrol agent BCA. The most commonly studied PGP are the Bacillus amyloliquefaciens, Bacillus lichenoformans, and Bacillus subtilis. The most common direct mechanism of the PGPB phytohormone production instead such as the phosphorus and nitrogen. The control of the pathogen various means, example hydrolytic enzyme, antifungal component, lipopeptide or antibiotics. Indirect mechanism of the PGP include production from the abiotic stress about drought, salinity, etc. Triggering of the specific pathway, particularly induction of the systemic resistance against the pathogen and pest, and release of the volatile organic compound. The Bacillus subtilis also used on plant as a fungicide. The bacteria Bacillus subtilis colonized on root system, compete with disease causing fungal organism. The Bacillus subtilis used as a fungicide fortunately does not affect the human. Bacillus subtilis bacteria secrete enzymes such as the amylase, protease, citinase, xylase and the lipase. The enzyme citinase is a highly involved in a biocontrol of the plant disease. Citinase is a most common enzymes in Bacillus subtilis. Bacillus subtilis became the attractive the biocontrol agent due to ability to produce the odd, the resistance endospores and antibiotic which control broad range of the plant pathogen. Bacillus subtilis with other organism by producing the antibiotics either kill or reduce growth rate. In this image you see about first crop health care. Bacillus subtilis used in a crop health care. Next agriculture pollutant, and then aquaculture, and last livestock farming also used. Bacillus subtilis was reported to induce the SAR, systemic acquired resistance against the bacterial pathogen, whereby the plant defense or trigger prior to the pest incursion. In addition, the Bacillus subtilis hinder the spore germination in a plant pathogen and the prevent pathogen from attaching to the plant. Biocontrol agent worked on a Bacillus subtilis against plant pathogen and the plant disease. This some recommendation crops are cucumber, eggplant, tomatoes, chili, cut flower, orchard, ornamental in greenhouse and the lawn and nurseries. These are recommendation crops for Bacillus subtilis. Target disease or Phytospora species, Aspergillus species, Pythium species, Rhizocortina species, Fusarium species, Butarius cinerea species, Sclerotinum rolsifi species, and Eustylogo species. Isolation of the Bacillus subcells. Bacillus subcells are isolated from soil sample by the serial dilution method, either spread plate method or pour plate method. Common culture medium used isolation of Bacillus subcells is Bacillus differentiation agar. Microscopic characteristic of the Bacillus subcells. First one, gram staining. Bacillus subcells was observed violet color, rod shaped, gram positive bacteria arranged single pair or chain. Next, motility test done by the hanging drop method. This bacteria are motile like swarming motility. Culture characteristic of the Bacillus subcells. Bacillus differentiation agar recommendation of the Bacillus subcells bacteria for differentiation between a Bacillus cereus and the Bacillus subcells based on a mannitol fermentation. Yeast autolysis provide necessary nitrogenous source of the growth of the Bacillus subcells. Magnesium sulfate and potassium chloride support sporulation, and ammonium phosphate maintain a buffering the action. Bromoglycerol purple act as a pH indicator to detect the mannitol fermentation. In Bacillus differentiation agar, Bacillus subcells show yellow color colonies. About Bacillus subcells differentiation agar plate. In agar plate, you see yellow color colony bacilli are present in surface area. In this agar, bromoglycerol purple act as a pH indicator to detect the mannitol fermentation. That's why the colonies appeared yellow in color. Next, biochemical characteristic of the Bacillus subcells. Catalase test positive, oxidase negative, urease negative, indole negative, MR negative, VP positive, citrate utilization test positive, starch hydrolyzation test positive, gelatin hydrolysis test positive, esculin hydrolysis test negative, OF test positive. This all are biochemical tests of Bacillus subcells. Formulation of the Bacillus subcells. The commercial preparation of comes various forms such as the solid, liquid, powder, tablet, granules, ball agent, liquid suspension, and the soil emulsion. This kind of thing used in a agriculture field, used as a biocontrol agent against plant disease and plant pathogen.